Next paper is a role of biotechnology in vaccine development by Amishi Gupta. She is from GBM Global School. JBM, sorry.
I'm sharing my screen.
Yeah.
Hi everyone, my name is Amishi. So my topic is biotechnology advancements in modern vaccine platforms. Now let's talk about revolution of vaccines. The COVID-19 pandemic sped up vaccine innovation like never before. But our longer research for an HIV vaccine shows we still need new ideas. This review looks at three biotech platforms: mRNA, viral vector, protein subunit vaccines to see how they work and how effective they are. The pandemic proved that biotech can make safe vaccines fast. Now the question is, which platform will be best for the next health crisis? Now let's move on to the next PPT. Evolution of vaccine technology. Ma'am, can you share the previous PPT? Evolution of vaccine technology.
Actually, your PPT, you have put some seconds there. Wait.
Yeah. Evolution of vaccine technology. I have represented this in a form of flowchart. Traditional vaccines, the foundation. Well-attenuated vaccines, example MMR. Inactivated vaccines, example polio. Toxoid vaccines, example tetanus. These are safe and effective but slow in production. Modern biotech vaccines, the revolution. Recombinant vaccines, example hepatitis. Viral vector vaccines, example AstraZeneca, COVID-19. mRNA, DNA vaccines, example Pfizer, Moderna, COVID-19. Designed in computer using recombinant DNA, synthetic biology, and nanotech. Now let's move on to the next slide. In the next slide, I have compared and shown the three champions of vaccines. mRNA. On the left, we have mRNA, the genetic message. It delivers a tiny strand of genetic instructions wrapped in lipid nanoparticle, piece of virus so the body can learn how to fight. Now, in the center, we have viral vector, the Trojan horse. This method uses a harmless adenovirus to carry the genetic code of the spike proteins into our cells, helping the immune system practice safely. Now, on the right, we have protein subunit named precision keep. Instead of giving instructions, it directly gives the immune system a purified piece of spike protein, allowing a very targeted immune response. Now let's move on to the next slide. Now we have the first vaccine, mRNA vaccine, the genetic message. This is the latest vaccine. mRNA vaccines work in a very smart and a clean way. Firstly, nanoparticles enter the cell carrying the mRNA safely. After entering the cell, the particle opens and the mRNA is released inside the cell. The cell's riboscopes read this mRNA just like they read normal messages. After reading it, they start making spike proteins. These spikes are harmless, but they help immune system practice. When the immune system sees these spike proteins, it learns to recognize and attack them. This prepares the body to fight the real virus quickly and strongly. The mRNA vaccines like Pfizer are known for being fast to develop and highly effective. But the main challenge is their cold storage. I have represented this whole process in the form of flowchart with images and text. First, you can see the lipid nanoparticle is entering the cell. After entering the cell, the mRNA is released and read by ribosomes. After reading, the spike proteins are produced. After production, the immune system recognizes and attacks these spikes. Now let's move on to the next slide. Viral vector, the Trojan horse. Viral vector vaccines work like a Trojan horse. A harmless adenovirus enters the cell, carrying an important genetic instructions. Inside the cell, it delivers the spike protein. The cell reads it this code and produces the spike protein, which alerts the immune system. This helps the body how to learn to the real virus. Some example of viral vector vaccines are AstraZeneca and Johnson and Johnson. They create a strong immune response, but pre-existing immunity to the vector and low effectiveness. I have also represented this process in the form of flowchart with images. You can see an harmless adenovirus, the vector enters the cell. It delivers the genetic code for the pathogen's protein. After delivering, the cell produces the protein, triggering the immune system. Now let's move on to the next slide. Now, this is the third last vaccine. The protein subunit vaccine. By giving the body only a purified piece of virus, usually the spike protein. In the animation in the side, you can see these purified proteins being injected. An adjuvant with a small boost symbol is added to make the immune system react more strongly. Once inside, the immune system recognizes the protein and builds the targeted protection without any live virus. The common example of protein subunit vaccines is Novavax. These vaccines are stable, well tested, and usually cause fever as side effects. But they are slower to manufacture and often need a booster to maintain strong immunity. I have also represented this process in the form of flowchart. Purified spike proteins manufactured in lab using insect cells are injected. An adjuvant is added to alert the immune system. Immune system directly targets the protein. Now let's move on to the next slide. I have compared the three vaccines according to their platform, mechanism, efficacy, stability, speed, and limitations. First, platform protein subunit uses purified viral proteins to trigger an immune system. Viral vector uses a harmless virus to deliver the genetic code of the targeted virus. mRNA uses messenger RNA to instruct the cells to make viral protein. Mechanism protein subunit. Immune system recognizes the protein as foreign and produces antibodies. Viral vector. Vector carries DNA of the targeted virus. Body makes viral protein immune response. mRNA.
Excuse me, Amishi. Please conclude your presentation in another 1 minute.
Okay, ma'am. Okay, sure. Ma'am, can you move on to the next slide?
Yeah.
Now I have represented this in the form of flowchart. Lipid nanoparticles for mRNA, vector engineering for viral vector, novel adjuvants for protein subunit, and an AI nanotech chip. Ma'am, next. Now let's talk about the future developments of scientists. Personalized mRNA vaccines, custom made to fight diseases like cancer by training immune system. Next-gen platforms, DNA and self-implying mRNA. New vaccines offering stronger and longer protection and faster production. Universal vaccines, a single vaccine can protect against the multiple viruses like flu. Personalized and preventive medicine, a new era focused on individual based treatments and preventing diseases before they appear. Now the next slide. Ma'am, can you move on to the next slide?
Yeah.
Thank you. Thank you, Dr. Pooja Bachani, my mentor, and thanks to the Global Research Hub. Thanks to the ICONSTARS for this golden opportunity.
So, Amishi, you're in class 7, right?
Yes, ma'am.
I really enjoyed your whole presentation, You are the youngest researcher to be future researchers. I really enjoyed your presentation. The way you I mean, your presentation was really very good.
Thank you so much.
But yeah, and I really enjoyed, seeing the way you delivered it, and the way you were speaking throughout the slides. It was nice.
Thank
So, shall we proceed and ask some questions? Are you ready for it?
you. Yes, ma'am, sure.
Yeah. So, I first of all, I just want to ask this very simple question: that what inspired you to choose this topic? Why vaccine?
Ma'am, because some vaccines are still not invented for the uncurable diseases like cancer and HIV. So I studied the topic of vaccines to understand like how the complex system makes it works in the body of human.
So, can you explain in just a simple words that how a vaccine trains our immune system? Just in a simple word, how it trains our immune system. What happens when you
Okay.
inject a vaccine?
Ma'am, basically it carries some harmless adenoviruses which is injected into the body, and it gives the messengers so the body reacts according to that. And when actually the real viruses enter the body, it is easy for the immune system to fight with that.
I mean, just give me an example of any virus you like.
Coronavirus,
Uh,
COVID-19.
okay, so what happened in that? We had a vaccine, right? So what happened in that? They
Basically,
injected a vaccine
ma'am,
on us.
the before for the prevention, vaccine is injected before the virus, and the immune system basically experiences and learns how to fight a new virus because the vaccine carries an adenovirus, which makes the immune system get trained. And when actually the real viruses enter the body, it is able to treat it in the right way.
Hmm. Okay, great, great, wonderful. You have answered it nicely. So there is something like memory is built in the cell, right? And
Yes,
in the
ma'am.
body, and then they recognize it. Yeah. Okay.
Yes, ma'am.
So now you are going to be a good researcher, Then I just wanted to ask one thing. While you were researching for all these things, what challenges did you face? What problem did you face?
The problem, man, basically the problem is like I have to collect, like I was not able to understand the three layers first because there are three layers of protection. So first it was a really complex task for me to understand, but gradually I learned the all the three protection layers, so I was able to understand.
Yeah, and the credit goes to your mentor, right?
Yeah, yes, ma'am. She helped me a lot. My mentor.
Great, great. Yeah, great going then. Okay. Thank you so much,
Thank you, ma'am.
and all the best. Okay.
Thank you, ma'am.
Thank you, Amishi. It was wonderful. And
I have one question. I have one question.
yes, sir.
So now, after going through this process of doing research on vaccines, is there a next research topic that you have in mind?
Yes, ma'am. Yes, sir. I have about the dermatology. I am interested in the field of that. Like, yeah.
Okay, what in dermatology?
Basically, it talks about the skin problems people usually face now onwards, and like the products which are recommended to use in such problems.
Okay, and what is the motivation behind wanting to pursue this research endeavor?
Because actually, I see many people having like many problems, skin problems. So they do not know the perfect treatment, how to deal with it. So I was just interested, like what products or things we can use to treat their skin problems. I see people around me who are facing these problems.
Okay. Yeah, yeah. Thank you, Mishi.
Thank you, sir. Thank you.
Okay. Thank you, Amishi. Yeah.